So now in the session last, but not at all least, is Benedikt Brors' Bioinformatics concept for the molecular tumor board.
Okay, so first of all, let me thank the organizers for inviting me here to this fascinating symposium and giving us the opportunity to present some of our work. As you see from the title and also from my affiliation, I'm slightly turning the focus a little bit towards bioinformatics, which is actually the underlying algorithms to call all the variants from which these fascinating findings have been made up. But I will also show you how this is being used in clinical decision making. I'm also indebted to the previous speakers, especially to Garrett, to introducing the topic to you, so I can be pretty fast in the introduction and skip over some things. Just to remind you, that's all in the context of precision oncology, and the way we understand precision oncology today is that we use some information from the molecular setup of a patient or from a tumor to inform therapy, diagnosis, also prevention strategies. At the moment, we're mostly using somatic mutations, and we're also using mostly single biomarkers to inform us about which treatment should be appropriate. That's something that's going to be changing in the future for sure. We can also use some transcriptomic information that's in clinical diagnostics still quite scarce. And in the future, eventually proteomics or metabolomics may be used as well. So all this has been only possible because we have an incredible increase in the throughput and capabilities of sequencing technology. And just list here some of the machinery that's available here at the Cancer Research Center with one HiSeq X10 cluster, 6 HiSeq 2000, 2500 machines, some smaller MiSeq sequencers, and a number of assays that can be run routinely, which have a nominal capacity of about 20,000 30x covered human genomes per year together. That's quite a lot to do. A lot of this is, of course, in the research context, but some of this is also being used in clinical diagnostics. We also collaborate heavily with the Institute of Pathology here at Heidelberg University Medical Center, and I will explain you this is quite important because they do validation for each and every of the informative findings with some technology that is accredited for diagnostics use. Okay, just very briefly, what do we do in cancer genome sequencing? Actually, what we do is we take the genetic material of a cancer cell and we cut it to very small pieces, basically in a big shredder. So this is like taking the Encyclopedia Britannica and cutting them into maybe centimeter-sized pieces. And then we use the computer to puzzle it all together. And fortunately, we have some blueprint, which we call the human reference genome sequence here, which is in the database. And we line them up at the position where they match best. And then we look for differences to the reference genome sequence, and this can be a single base exchange here, so a point mutation or a small insertion deletion, or it can be gross things that happen in cancer. So certain parts of the genetic material may be missing completely, or just one of the two alleles is missing, or we have some amplification of genetic material. And we can have rearrangements like these translocations here. So these things can be pretty easily be seen. However, there are a couple of challenges here. You know that cancer doesn't have these nicely ordered 46 chromosome karyotypes. This is a picture, multicolor fish picture from acute lymphoblastic leukemia, where you see sometimes you have 3 chromosomes, sometimes you have just 1. This chromosome is made up of parts of other chromosomes. Here you have 3 different chromosomes stitched in together, and this is what makes life actually hard, and it's not nicely lining up across the reference sequence. But we have some methods to actually get this information somehow. There's also the problem that the cancer samples that come from a routine surgical theater are almost never 100% tumor cell. content, but have various admixtures of different other cells, and this dilutes the somatic mutations. I should also mention here that we do the very same with non-malignant cells from the very same patients to rule out that the base exchange here, for example, is a polymorphism. If it is not, we call it a somatic mutation, so it only occurs in the cancer cells. Heterogeneity is another problem. Cancers are made up of different parts that harbor different genetic alterations, and polyclonality can be there. The logistics of the entire workflow is quite ambitious, and there are also bioethical problems because we have the full genetic information of a single individual. We need to protect this, of course. Just to give you an example of how complicated this can look, these are medulloblastoma cells here, and one of these medulloblastoma cells is harboring a high number of extra chromosomal elements which contain some oncogenes from an event called chromothripsis. All this is a computational and IT challenge. We process quite a number of data here for different types of analyses. At the moment, our center is producing approximately 3 petabytes per year on new data. And of course, we also need to store the old data. That's a problem of storage systems. They never will be freed. So this is quite a high operational cost for them. We have roughly 4,000 compute nodes available. And we also have occasional problems with our network infrastructure because we need to transfer the data even between different rooms is sometimes a problem. So this, we're working on that. It's not hopeless, but it's also sometimes challenging. Over the years, we've developed a quite nice pipeline to call different types of alterations, starting from DNA and RNA sequences. A number of people have been involved here. So there's a primary analysis that's mainly the alignment, so this puzzling together, the lining up across the reference sequence. And then a couple of quality control steps, and we'll go a little bit into that. Removal of duplicates, merging different lanes, calculate coverage, match genotypes between tumor and normal cells, and so on. The real interesting part is here, where we call different types of mutations, the single nucleotide variants, copy number variants, structural variants, small insertions, deletions. We also check whether mutations are there in RNA sequencing data, so whether the variant is expressed. And we deeply annotate the mutations, and this proves quite to be important. I will come to that later. And in the research context, we've developed a number of alternative procedures here. I will not go too much into detail, just focus here on this last one. This is actually the different types of base exchanges or point mutations that can occur. So there are 6 different types that can occur in principle. And if you take the base just before that and just after that into consideration, there are 96 different types of the mutations that can occur in the human genome. And this is a typical picture. You see that C2G mutations in a CpG context are the most common ones. They are also the most common ones in evolution. That's due to deamination of cytosins. But we have different types. And nowadays we can actually computationally decompose these spectra into different what's called signatures. And they tell us something about the mutation-generating mechanisms. So there are signatures due to tobacco use. There are signatures due to deficiency in BRCA1, BRCA2. There are signatures due to other oncogenes, to viral infections. And this helps us a lot to find out what's happening in these different cancers. That's quite exciting new technology that's available here. We've used that into different research contexts, and I'm also skipping very briefly over this just to mention 2 of the findings here where we actually contributed. Large part of the work has been done by the departments of Stephan Pfister and Peter Lichter here at the Cancer Research Center, of course. But in medulloblastoma, one of the interesting findings was that a high number of the mutations we find, and actually these pediatric tumors harbor just a few of the mutations in the coding region, they hit genes that are important for epigenetic modifications or chromatin maintenance. So like SMARCA4, this part of the SWI/SNF complex, MLL2 is the histone demethylase, KDM6A as well. And we don't know exactly what they're doing yet, but it seems to be that they have an importance because we find them quite frequently. And they show very high subtype specificity. For example, SMARCA4 mutations are only found in this Wnt tumors and group 3 tumors, and others in other subgroups here. So that's quite exciting. And in another entity here, pediatric astrocytoma, was even more exciting because all of the mutations were downstream of a single receptor, the FGF receptor 1 in this pathway, most commonly BRAF fusions here, but also others. And they show almost perfect complementarity. So mutations, cancers that have a BRAF fusion never have a BRAF mutation or FGFR mutation, and so on. So these are quite interesting research insights, but finally what we want to do is to go one step further. Before I come to that, I mentioned that quality control is actually very, very essential. There are a number of things that happen. Sample swaps happen if you run this in high throughput. Contamination with non-matched DNA happens, as has been set out before. Another problem is that sometimes we find tumor DNA in non-malignant sample, and these are usually blood samples. And you can imagine that just for biological reasons, there's tumor DNA in the blood because you're also doing liquid biopsies where we hunt for this tumor DNA in the blood. And they may be co-amplified or there may be cancer cells in between. And this is a problem for the calling algorithms, not to confuse them with germline polymorphisms. There are duplicate reads that come from the same PCR template. There are certain patterns like this GGNNG pattern here that causes the polymerase in the sequencing reaction to stall at one position and read one base false. This G is read actually when there's another base, it's read as a G. There are guanine oxidation artifacts that happen due to sample handling. There's a GC bias, probably I will skip to that. There are regions that are very hard to map because they are full of repetitive elements and so on. And this is quite some work to actually get rid of all of these artifacts and make sure that the data is high quality. I will skip over that. We've been benchmarking our algorithms, of course, in the context of the International Cancer Genome Consortium. And this is a paper that will come out in 2 weeks in Nature Communications. And it shows here the benchmarking in a large set of mutations that have been independently verified. So we can do this precision versus recall plots. Here's the precision, here's the recall. And you see here for the simple somatic mutations a nice curve as you expect that should look like this. And the best algorithms are in this corner here. We are actually capital letter I, so we are here. So we are not bad compared to others, I would say. For indels, somatic small indels up to 30 bases, it looks much worse. Almost everybody is somewhere. You don't see this nice curve here, and everybody has some problems. That's still unsolved issues. So we need to actually validate all the indel findings that we have in a clinical setting. Okay, now I'm coming to how we use that for clinical decision making. And this is the way it works. From a tumor sample, from patient we get a tumor sample, blood sample. We extract the germline DNA, tumor DNA. We also extract tumor RNA if possible. We sequence that at high coverage. We do the computational analysis as I've shown to you. And we get a spectrum of somatic mutations. So that's very much possible, maybe still incomplete. There are false positives in there. We have to take care of that, but it's possible. The really tricky part is here to get that into a comprehensive report that tells the treating physician something on how particular patients should be treated, what the best therapeutic options are, maybe in which risk group a patient is, whether there are mutations conferring resistance to existing therapies, and so on. So this has been established at the National Center for Tumor Diseases here in Heidelberg in a particular trial, the NCT Master Trial for adult cancers. We've seen now over the last roughly 2 years, I think close to 300 patients now, and the complete workflow in fast track is about 3 to 4 weeks. The oncologists are telling us that's still okay. The recent findings is that 62% of the patients have potentially actionable findings. I have to say this is a selected cohort with a lot of young cancer patients, so this is an unusual composition with respect to cancer types. This may not be representative of the entire cohort of cancer patients. We do the validation by certified techniques, mostly Sanger sequencing, sometimes PCR for fusion genes, or immunohistochemistry. And many of the findings we have are not typical point mutations. These are the ones that you can also detect by panel sequencing, for example. But they are copy number variants, structure variants, or indirect, like upregulation of a ligand to a receptor tyrosine kinase. I have to mention this is exome sequencing at the moment, so we sequence only the protein coding part of the genome. We're currently transitioning into whole genome sequencing due to the availability of the technology now, and also it's getting more economic to do that. That's the entire workflow. It's quite ambitious. It involves things like having a informed consent from the patient in the right format that allows us to do all these things, also to do research on the data. It involves quite a lot of quality control on the side of the pathologists. The sequencing and bioinformatics, that's the most challenging part, the clinical interpretation. I will come to that. And then the validation of the immediately actionable lesions by certified technology. And then finally the molecular tumor board and eventually the treatment here. This is a report form that we use in tumor board discussions for these patients. That's a single example from a patient with mammary carcinoma. We report the total number of SNVs. That's quite low for breast cancer, but still in the range of what we find. You see that here on this bar as well. And the different single nucleotide variants. You see some cancer genes here, like SARC, but this is a variant of unknown significance, so we don't know whether it has any functional impact on the protein. This mutation is found in cosmic, so very likely to be inactivating to TP53. And there are a number of copy number alterations. And what's quite interesting here is that we have multiple amplicons, including PDGF, alpha, EGF receptor, FGF3, FGF4, and cyclin D1, plus some fusion here. And we also see high expression of some of these genes that are here. That was quite interesting. And the way this is being interpreted by the oncologists, which takes about 5 to 8 hours at the moment per patient, is that they see, okay, there's an amplification here of this protein. This is an inhibitor which is deleted. And we have this high expression as well. And this then leads to a hypothesis that blocking or taking also here FGF3 or 4, which are ligands to this receptor, which is unaltered, by the way, are there, that pan-tyrosine kinase inhibitor like lapatinib may be useful, or alternatively, what is considered as also immunotherapy for this patient. This is some overview figure. The numbers are slightly outdated. I believe this is from April this year, where 269 patients had been enrolled going through that, and to tumor board 152. And these are the different baskets of therapy that we have here, like PI3 kinase, AKT, mTOR pathway, RAF-MEK-ER cascade, receptor tyrosine kinases, developmental pathways, DNA damage repair, and hypermutation. And you see also that some are preferably filled here across the tumor entities that we had. So what are the future challenges for this type of, let's say, genetically informed decision making? I have to mention these are patients with no further therapy options. One challenge in total, and this would also bring more patients into therapy, is to run clinical trials earlier on in cancer development where it makes sense and where it's ethically possible. And also run these clinical trials simply to provide a certain molecularly informed therapy to patients. Another one is that resistance. We see in targeted treatments a rapid onset of resistance. That's very typical. So a lot of cancers can very quickly get around this blocking of signaling pathways. And this one way to tackle this is combination treatments. And that's much more challenging to predict combination treatments than single ones. We have some ideas on how this could work, but it's far, far from being reality at the moment. Another thing is tackling intratumor heterogeneity. That's also another reason for resistance, that a very small proportion of the cancer cells actually has already mutations that make them primarily resistant against the therapy. And if you treat them with a particular drug, they will overgrow all of the other cancer cells which are killed by the drug. And the prediction is right now just matching with the database of drug targets that could also be improved in the future. We also probably need more systematic recording of mutations, of the therapies prescribed and the outcomes, and ideally also on an international level. There are now several efforts to do this. You may have heard about the AACR Genie database, where 6 different centers are working together to share their findings. And I think that's one way to address this. There are many more approaches outside, and I don't know who will finally make it, but I think we all will benefit if, for example, we find a particular genetic constellation which we've never seen, but it looks very interesting with respect to a particular treatment strategy, and we find out that a patient just 2 years ago had the very same constellation at Memorial Sloan Kettering Center, or in Barcelona, or in Singapore. And he was treated with a particular treatment with a fantastic success or with a complete failure. And as somebody consulting treating physicians, I would like to know that, to give them this information, because it's important. But we can only do so if we have this information. So I think the future, and this is my last slide actually, is in data integration with a number of additional things. So we have the mutational patterns. We want to look across how they occur in different cancer types, for example, to find out whether a particular variant is probably of significance for is a functional mutation. This is the case if it's also found in cancers with low mutation rates, how these mutations scatter across the protein sequence, whether they aggregate in domains or there may be hotspot mutations, how the success was with respect to therapies prescribed for different patient groups with and without mutations. And there are also in vitro data, and I'm deeply convinced that this will be crucial if we go to predicting combination treatments. We will need to have data from drug sensitivity screens on cell lines, patient-derived xenografts and others, where these combination therapies have been tested, because it's very hard to do this ab initio just without any knowledge. And also synthetic lethal interactions may be screened from drug screens, from microRNA screens and others. This data is around, and it's a huge challenge to integrate that, but there will also be huge benefits. Yeah, this work hasn't been possible without the contribution of many, many different people. A lot of talented bioinformaticians, of course, which are listed here in my group, in Roland Alt's group at the Cancer Research Center. And very crucial is the interaction with the oncologists here in Christoph von Kalles' department at the NCT. Sequencing core is important. There's the sample processing lab, pathologists, a number of colleagues from the DKFZ, a number of consortia where we have used data in research studies, bioinformaticians here with whom we are collaborating, and oncologists at the University Medical Center in Heidelberg. So that's it. At the end, thank you for your attention.
Thank you very much for this exciting presentation. Questions? May I start with one? Sort of the bioinformatics concept of then linking the trial experience, sort of, as we have heard in the previous talk. How do you see that happening in your concept? So at the moment, we're working on integrating data from the NCT Cancer Registry, which has the follow-up information on the patients that have been treated, and making that available. There are a number of different ways on we explore this. We have a collaboration with SAP on that to put this into a device where a lot of the information that we have on a particular patient is used. There are a number of challenges with respect to data protection, to data privacy, but they can be solved. However, I have to say it's hard work, because there's a large different number of scenarios how patients are treated. A lot of patients are treated outside of Heidelberg at practicing oncologists and other clinical centers, and following this information through is quite some challenge. That's probably one reason why having interventional studies with a tighter regulation on the recording of the follow-up would be probably helpful. Yeah, we certainly need that, because, I mean, finally, these treatments are very expensive, and to convince somebody to pay for that, like a health insurance, you need some data on a large number of patients to really show that you can make that this makes a benefit in the clinical care of the patient. That's the ultimate goal, right? Just showing that we have nice scientific hypothesis and we get stable disease for 3 months or so is probably not enough to convince somebody to pay 100,000 euros for therapy.
Rita?
Thank you very much, Dr. Brors, for this very comprehensive talk. My question is concerning tumor heterogeneity you mentioned already. With respect to the 60% of the patients where you detected druggable or druggable mutations, do you have any kind of cutoff up to which allele frequency of mutations you would advise to track the disease? Or is it just the detection or the validation by Sanger sequencing that means about a cutoff of 10% of mutated alleles? I guess there is a discussion going on in your group as well about this issue.
Yeah, you're absolutely right. At the moment, the cutoff is mainly determined by technical limits, also on how low we can go with still reliably calling a mutation and not a sequencing error. There are a number of heuristic rules that we have developed, and if you translate this, in most situations it goes probably down to 8%. That's very close to what we can validate also by Sanger sequencing. We had rare cases where we tried a very low allele frequency mutation to validate. That's a lot of work because you have to clone this mutation, then pick 1,000 clones and sequence them and so on at the particular site to find low allele frequency mutations with Sanger sequencing, but that's very hard. We're not sure also at which particular level something's still relevant. If it's a resistance conferring mutation, it's certainly relevant if it's they're only in low allele frequency, but there are technical limits from the typical coverage, which is about 100x we have in exome sequencing routinely. For the ones that are driver mutations, we're not sure whether it's relevant if it's only in a very small proportion of tumor cells. Then if we positively target only this fraction of the tumor cells, we'll probably not kill the rest of the tumor. That's the other side of the medal, so to speak. But at the moment, we're down at 8 to 10%, and we have to see. There's one interesting paper by Andrew Futria's group in Science, I guess, where they took different pieces of lung cancer and sequenced it. And of course, there were mutations that were only in certain pieces. But when they took one of the pieces and sequenced it to 800x coverage, they found all of the mutations. So that could be one way to address heterogeneity. At least the first hint, if you sequence very, very deep, you will find all of the mutations in that particular cancer. But at the moment, it's not affordable to do this in a routine setting.
Any further questions? If that's not the case, everybody's looking forward to the panel discussion, I guess. Thank you very much, Benedikt.